inflammation. I remember in lecture you said that they break.
Well, I don't know if they break or they just remove the proteins, into the cell.
Mm-hmm.
And a lot of times, I don't know the fact that happens in these cell junctions at that time, but they just take vesicles and put the tight junction into a vesicle so it can go in and out of the membrane.
Oh, that's weird.
Yeah, so I don't know if those particular cell junctions during that event do it that way, but some cells do that. They become, more junctions by inserting the vesicle as to part of the plasma membrane and then remove them by going back into a vesicle so you can quickly increase cell junctions or decrease them that way.
So is that a thing for like every vessel then, that they just have like tight junctions they can pull out when needed or put in when needed?
Again, I don't know for particular circumstances. I just know that cell junctions can be moved in and out like that. And so I don't know for every circumstance.
Okay, that's cool. Thank you.
Okay. All right, let me find our study guide. I think I have it in there. Okay. I have the picture. Maybe not. Okay, so I might have to. Oh, I do. Okay, great. All right, so let's see, we have about 17 people here. So what I was looking up while I was talking to Noah was the study guide, this second study guide. If you want to open that up, I'll share that with you guys, and let's just give it a minute so other people can jump in and kind of walk through this. So I am following along with the lab manual. It's just that I'm not sure why they maybe cost, wouldn't be surprised, that they took out some of the pictures, the photomicrographs of the actual tissues. So they have a high magnification, I believe, of just the lymph node, and they show a tonsil, which we don't have a picture of, but they got rid of they have a low and a high of they have a high magnification of the spleen and a low magnification of the lymph node, but they don't show the thymus and they don't show Peyer's patches anymore that they used to show. And I believe they have pictures of most of those in the textbook instead, but I'll walk you through some of those slides so you'll know as you go to draw them where to look. Okay, so let me share first the study guide with you, which you have. Oops. And of course, my dog wants to go outside. Okay. So if you look in the lab manual, let me go to that page, the very first figure they show you is this picture of a human, and this man's lymphatic capillary, larger vessel system that are collecting from the capillaries, and the location of lymph nodes along that passageway. If you go back to before the lab exam, which I know is like hard to remember, when we did the lymphatic system, the lymph is picked up from that extra 2 millimeters of mercury that on average leaks out on the arterial end and isn't brought back in on the venous end of the blood vessels. And instead, the lymphatic system is picking up that extra fluid and it has to return it to the bloodstream. So this is showing you not only how that happens, but the location of lymph nodes that are there to filter that lymph in case it picks up pathogens or bacteria or fungi or viruses along the way, and to trap those things. So here you have a few lymph nodes kind of in the lower part of your upper arm, but most of them are in the axillary and the thoracic region, the cervical region. You have quite a few around your respiratory tract and your oral cavity. Not so many up when you get into the out here at the extensions of the skull. So around any kind of mucosal membrane, you're going to see lots and lots of lymph nodes. So often if you get a cold or you have an infected tooth, you'll notice enlargements of these that you can actually feel. They get so big because you're going to learn shortly that adaptive cells are undergoing mitosis rapidly within them, and they're actually literally growing, and then they shrink back down as those cells disappear and you're better. So all along your mucosal membranes, you're going to see lots of them, so lots in the groin area. Now in the legs, they don't show the legs here. You have, like the arms, very few lymph nodes. So the massive numbers of them are going to be in these inguinal areas to filter the draining lymph from the legs. So know these locations using landmark terms like inguinal, axillary, cervical, and vertebral areas here for the, and of course the thorax and the abdomen. The other thing to note, and the color coding here is showing you this, that the lymph, we're kind of asymmetrical, that the lymph that is draining from all your legs, your abdomen, and much of your thorax, although not all of it, and your left arm, left shoulder, left side of the head, all goes enter back into the blood into the left subclavian jugular junction right here, called the thoracic duct. Your right equivalent thoracic duct is only getting from your right head, neck, thorax, and right arm. So much more lymph goes into the left thoracic duct. So why that is probably because of a structure we're going to look at in the models right here called the cisternae chyli. I know it's an awkward name, but that's what they called it here. So you can see all these vessels coming, not just from your legs and your arms, but many of the abdominal ones ultimately go through this junction point, like a big highway intersection called the cisternae chyli, and then from there joins into larger ducts that eventually head up into your left thoracic duct and enter back into the blood. So understand the green versus the reddish brown colors here and that asymmetry. So they have a higher magnification picture of this structure. Here's the cisternae chyli with many of the lymphatic vessels converging on it. And then this whole long, large duct that's leaving it is all called thoracic duct, including all the way into where it dumps into the left side here. This is all still called thoracic duct. And there's the jugular and the left subclavian veins. And that's where the lymph is going to enter into that. So you'll see it quickly goes into the heart. So you want to make sure you've got it well filtered along the way. These are, in this figure, these green things are lymph nodes, but over here, we'll see in a second, and I'm sorry the lighting wasn't a little bit better in this photograph, but we'll look a little bit closer at our model here. And then on this side, you're seeing the right thoracic duct, which of course is just coming from the right side of the head, the neck, the thorax, and the right arm. So on this picture, and what did I do with this picture? Oh no, this picture isn't a good view. We'll have to go into our actual photographs. You have the thoracic duct here. because the heart's missing in this model, but there is a cisternic hilate. I must have cut it off when I cut and pasted this picture into here. Sorry about that. Maybe I'll have to edit that and make sure I get the cisternic hilate, because it does show up. So this pink is all thoracic duct, and you just have to know it's going to go into the left subclavian jugular junction, because you can't see it when the organs are removed. Now, don't get confused by these pink lines that are coming off here with little bulbous things on them. They are not lymph nodes. These are actually meant in our model. This chain here is meant to be the sympathetic ganglion chain, and these are sympathetic nerves. But so they don't show any nodes on this, just the duct. They do show the cisternic hilate though, so maybe I will exit this share and go into your photographs of the models, lab 2 test models. This is where I copied this picture, but I must have cut off the cisternic hilate inadvertently. Okay, so hold on and I'll find that. What did I do? Put it in the, yeah, so I put it in a labeled PowerPoint. It's in your model one, so you actually get it all labeled. Let me share this screen with you. Kind of like the blood vessels were labeled. And it's the only model we have of the lymphatic system. I have to try and drag this up a bit. So here the pink is the thoracic duct. Let me see if I can zoom a bit. There's the thoracic duct. These little knobbin things are sympathetic ganglia, so you're not labeled here. They're not part of the lymphatic system. And here is that cisternic hilate where all these ducts are coming together. It's a junction for a lot of lymphatic vessels. These pink, just so you don't think pink is anything, these pink is always lymphatic. These pink are actually parts of the ureter. So we'll come back to them in the urinary system. And all of these pink things are meant to be sympathetic nerves. So the only parts you need to know for this are these 2 parts, the thoracic duct, cisternae, hilate. Is that clear on this? Any questions? Let me stop sharing this. Any questions on that? I don't see anything in chat. Okay. So you have a labeled model picture. It's the only labeled model picture that you need. So let me go to exit this and go back to the study guide. Because this is another source of your photographs. You go look in the folder of the histology though. But before we get there, let me share this screen. And this is again out of your lab manual, so you can just read about it there. I also wrote stuff here. There is a picture, and I'm just moving it over so guys aren't in the way, of this fellow here showing you who are the lymphatic organs. So we don't have any histology slides that are very good for the tonsils. You probably already know you have tonsils in the back of your oral cavity. And these have crypts in them. We talked about crypts where you're cultivating microorganisms. There's a nice picture that histology in your lab manual. So I may add that as just a good way of pointing at crypts. Crypts are also present in the appendix down here, although I don't think there's any picture of those in your textbook or lab manual. So you're going to be drawing the thymus here. Now the thymus is most important in activating your adaptive and selecting proper cells for your adaptive immune response. So we're going to hear more about that next week. your bone marrow, which of course makes the white blood cells, and the spleen, where you're going to filter blood. So you're going to have lymphoid tissue. It's not a lymphatic organ, although the spleen is sort of considered a lymphatic organ because you could dissect it out. But most of the spleen, the vast majority of it, is recycling red blood cells. There's just these little islands that look a little bit like a Peyer's patch in a way, but now embedded into the spleen, which is called white pulp, which we'll see in a minute. The lymph nodes are not shown here, but they are a lymphatic organ. And then you have Peyer's patches, which are lymphoid organs. And they are all over your small intestine, but there's a huge cluster that you can actually see on the slide with your naked eye. It's so big, called Peyer's patches. And that's right before your small intestine goes into your large in the ileum. And they're probably so enlarged there because you have so many microorganisms in your large intestine that you need a lot of lymphoid tissue right at that junction in case anybody escapes upstream and you could potentially get infected. So you're going to draw all these histology slides, which I believe there are 4. There's Peyer's patches, thymus, spleen, lymph node. There's 4 of them at 40x. But then you're going to draw some of them again at higher magnification to look for specific structures. So why you're drawing them all at really low magnification is because they're all made of the same reticular connective tissue. So if you go really high magnification, they're all going to look kind of the same. So we have this hematoxylin and eosin stain on all of them. So unlike the spleen that you drew in 103A where you saw the reticular fibers, none of them are stained to show it's reticular connective tissue. You just have to know that it's reticular connective tissue. So it does tell you that. Now there are lymphocytes and dendritic cells and macrophages found within the lymphatic tissue, reticular connective tissue, because that's why you want to use reticular fibers, so these cells can sit on there and wait. So you're going to be drawing them all 4 drawings then at low magnification. Now, which ones do you have to go to higher magnification? Well, for the spleen, I want you to go to higher magnification and draw the red pulp, the part that's recycling red blood cells. And it's got sinusoidal capillaries. So because they're sinusoidal capillaries, they call the empty spaces, there's no blood in this particular animal spleen, they call the empty spaces in the red pulp venous sinuses. So remember, sinuses are larger diameter vessels, and this is capillary actually, not a vein, but it's collecting blood, slowing it down so that the macrophages that sit on the outside of the endothelial lining can see if there's any old, dead, or dying red blood cells, or if something is going through the bloodstream, which is a pathogen, a bacteria or a virus or fungi, it can also grab those. And when it does grab them, it's grabbing them on what they call the splenic cords. So the cords are like the frame of the, I'll point it out in the slides, but the frame of the picture being that blank empty space where the blood would be. But the endothelia and their associated macrophages, probably dendritic cells as well, are all sitting on the endothelial wall on the tissue side, and that's called, those 2 cells together, the endothelia, and the phagocytic cells are called the splenic cords. So you're going to draw that at high power because you wouldn't be able to point at things, and I wouldn't be unless you go to high power. So that's in the spleen. You do not have to draw the lymph node at a higher power, but if you wish to, I may end up showing it at a little bit higher power just to be able to put the pointer point at something and, very specifically, but you don't have to. That's optional. And then the thymus, you need to draw at higher power in order to see something in the medulla called the thymic corpuscles. These thymic corpuscles are associated with the development of a particular lymphocyte called T regulatory cells that we'll talk more about in the adaptive immune response. The thymic corpuscles, the biggest mistake people make is confusing them with a blood vessel. If there is a hole in the middle where blood would flow, that's a blood vessel. Thymic corpuscles are solid, but both blood vessels and thymic corpuscles stain very lightly in this. So you want to be at high magnification to be sure that's what you're seeing is a thymic corpuscle. So before we go here, let me go and I'll show you the slides that you're drawing. So I'm going to exit this again. Here we go. And go to the histology. So you can see there's quite a few slides here at different magnifications. So if we take a look at, I didn't identify which organ it is, so I'm not going to do that because I want you to be able to do it on the test. So if we take a look at this, and what you're seeing is a darker rind, that darker area has a higher density of cells. That makes it the cortex, and that's going to be a general theme if it's an organ like this one. Now, how do you know it's an organ? Because there's tissue on the outside here. It's very light at this power. You can barely see it. That is the capsule around the outside. It's loose connective tissue, especially in this particular organ. And then the lighter middle area is the medulla. So when you see more than one cortex around a medulla, cortex around another medulla, and you see these long, thin extensions of capsules called trabeculae going into it, it's going to be the giveaway that this is the thymus. So the lymph node, which could look like this, is only going to have one medulla, and the rest of it is a capsule. You're not going to have multiple ones. It does have trabeculae, but they don't go in as far as this one. And the other giveaway are these structures, which in this case, these two are staining pretty darkly. That's a thymic corpuscle. So let me close this one and find one at 400. Is this the correct one? Nope, that's not the correct one. Did I do one at 400? Maybe I only did it at 100 for this. Guess you'll have to do it at 100 if I only did it at 100. I can't tell from this picture. I'm not sure any of these have the thymic corpuscle, so maybe I'll have to amend that. Yeah. Well, let me go on here. This has got, you can only see part of it, but this has got one cortex, one big medulla. The other difference from the thymus is this medulla looks kind of airy, like little patches or streams of clustered cells and then little spaces. This is a lymph node. Here you see the capsule much clearer too on the outside. It got ripped off here. And the trabeculae occur, but they kind of just indent just a little way so that the outside looks like it has bumps on the outside. So this is the medulla, and here you have macrophages and T cells. Now, why there's so much space is so that the lymph will pool inside of this and allow those macrophages and T cells to potentially phagocytize something or encounter an antigen. When they do, they're going to go out to the perimeter here into the cortex and look for B cells. So out in the cortex here, we're going to have B and T cells. And if you're seeing like here, you don't see this dark ring with a light center or a dark ring here with a light center. This is what an unactivated part of the B, these would just be B cells waiting for their antigen. These areas have B cells becoming plasma cells. So these areas where you have the light center and the dark outside, these are called germinal centers, where they're plasma cells secreting antibodies. So those antibodies are going to go into the lymph here and leave the lymph node and ultimately enter into your bloodstream. So this particular animal had quite a few germinal centers. Here's one here, here's one here, here's one here, here, and there's one. Just a few areas where you don't see it. So my guess is they probably exposed the animal to something, so they'd get a lot of germinal centers. This, I'm sure, is not so this is the spleen. Now, again, it's going to look like one cortex because it only has one cortex. And this does look a bit airy, but this is such a huge organ. I mean, I think this is a human spleen. And it's like the size of your fist, or depending how big your fist is, like a man's fist. It's big. So you are going to see it curve at all. It's almost going to look, it's hardly curved, versus a lymph node, if we go back. It's a fairly small thing. So these are the same magnification. You can see the curvature is much steeper because it's going to, if you envision the rest of it, it's going to wrap around and be smaller. So at low power, to know this is the spleen, you'll notice the cortex is dark again, very thin. You don't see germinal centers because they don't occur in the cortex. You see a very thin rind of the capsule, and then you're starting to see different appearances in here. We need to actually look at another picture of this. That's another lymph node. You can see the germinal centers. The germinal centers are in the cortex for the lymph node, and they're not in the cortex for the spleen. So this is a really low power of the spleen. Again, here's that capsule. Here's that thin cortex. The other one was at 100x, I think, and this one's at 40. And then this is a germinal center in the white pulp. See that little crack there? That's where blood would flow. This is a blood vessel. This is a germinal center. We only have one germinal center. This is lymphatic white pulp without a germinal center. See the opening there? That's a blood vessel. Here's another germinal center free white pulp. So that's white pulp, white pulp, white pulp. And you will notice the white pulp's often around the larger blood vessels, are often, not always, but often flowing through them. This is probably another larger blood vessel. So what's red pulp? Well, everything else is red pulp, all of this. So the vast majority of the spleen is made of red pulp. So red pulp's where those red blood cells are recycled. So you're going to go higher. We'll look at it 400 in the area of the red pulp to see the venous sinuses and the splenic cords. And that must be this picture. So this is red pulp now. And these empty spaces are the sinuses where the blood would flow. And all we are nuclei stained here, but you can see the nuclei along the edge. And some are not very flat, so I wouldn't ask you, but my guess is those are probably macrophages. Whereas some are much flatter nuclei, those are probably the endothelial cells, like here, lining the edge of this capillary. So these things right here are called splenic cords. They actually form sort of the framework. And there is reticular connective tissue out here that's not part of the cords. This is the splenic sinus. So try and understand the difference. So anything that's right along the edge of that opening is a splenic cord. And that's where you're going to find those dendritic and macrophage cells, as well as, of course, the endothelia that make the wall of the vessel. So you might say, well, what are the nuclei in the middle here? They're probably fibrocytes making the reticular connective tissue. If there are questions, let me make sure I can see the chat at the same time. More, I have to hit the more. Doesn't look like there's anything in chat, but I'm kind of leading you through these slides. Okay, so I skipped this one. So this is, let me get rid of chat, which is now in the way. This is a low magnification picture, and it looks like it is also the spleen. There's just so much of this here, and then that looks like lymphatic tissue. So one of these must be these must be the Peyer's patches. Yep. So this is Peyer's patch. I know it looks like Peyer's, but they're British, so what can I say? It's Peyer's patches. Or Peyer's patches, not Peyer's patches. And this is the smooth muscle of the intestinal wall. And so this is an area called the submucosa. We're going to soon learn the layers of a mucosal membrane. And then hard to tell here because it's a bit cut off. But these are the big folds that make the epithelial lining. So these are little simple columnar cells. They're just folded back on themselves very much. So, well, you wouldn't call it food by this point. It's in your, end of your small intestine, but that would be flowing through here. And then this is the absorptive layer. So if you're absorbing anything from your ileum at that point, maybe water or some vitamins, or there's not much left to absorb at that point, but if you are, then you're going to be absorbing it right here. This is epithelia, and underlying it is areolar connective tissue with a lot of capillaries. So you want a lot of immune cells to be located right near that absorptive layer in what's called the submucosa. So these are almost like, if I could cut out and insert into the submucosa, part of the cortex of a lymph node. So you're going to have T cells and macrophages and T cells are probably busy here in the connective tissues here. And then this would be B cells that have been activated. So this is a germinal center again. Doesn't have a complete circle, but has a dark edge, and then plasma cells would be being generated in this area. So all three of these patches, these lymphatic nodules, which by the way, there's kind of two terms for these circular components, nodules and follicles, and they're used about 50-50 from what I can see in the book, but they mean the same thing. So one of these bumps is a follicle. Same with if we go back to the lymph node, they're going to, right, this is the lymph node. Oops, nope, that's the wrong one. The lymph node here. Each one of these bumps is called a follicle again, or a nodule, again, about equally. They're synonyms for each other. So I think, there's our 400. I think I mislabeled this. This is 400. That's a thymic corpuscle. So I think I said 40x here. This should be 400. So let me see if I can rename that so that's your 400. I don't want to move it. I must hit the wrong thing. I want to rename. This should say 400. Okay. Now you can have you can draw the other 400 level picture. Okay, so I am going to, if there aren't questions, let me go back and check that no questions. There is, it looks like something showed up. Can you point out the B cells? I can't really point a B cell out, but I can go into the Peyer's patches here. And this light area is a germinal center. So you would label that germinal center, and then you just know that B and T cells would be found in here, as well as plasma cells. So if you're actually seeing this light center, that means plasma cells are being generated from the B cells. All right, any other questions before I turn you guys loose? Just wanted to make sure what we had to draw and label. I think that's all listed for you in the study guide. So you're labeling, germinal centers, cortex, medulla, venous sinuses, kind of the things that I just went through. So hopefully they're all in that study guide. Yeah. So if it's not, let me close this and make sure it's in the study guide. Lab test. Sorry. Somebody's calling me, doesn't know that I lecture at this time. And my phone is on the other side of the room. Well, I guess I could just decline. It's just hidden under you guys. I should probably give up in a second. Okay. Let's let this load. Yep, here we go. See? Here are all the things you're supposed to know about these guys. So I'll make sure I reread that it's all there. Locate the germinal centers, recognize Peyer's patches, the red versus white pulp, the capsule, cortex, medulla for the lymph node. And I guess I didn't put down germinal centers, but it did say know where the B and the T cells are. Here we go. Venous sinus, splenic cords, thymocorpuscles. I think that's everything that you need to label. Any other questions? Let me get back to chat just to make sure, because I keep making it disappear so I can read the slide. Okay. All right. So if anyone would like to be in a, let me stop sharing this. If anybody would like to be in a group and work together, then let me know and I will set that up. So just put that into chat. You don't have to say it personally, and I will set up some breakout rooms. Otherwise, I will turn you loose to go ahead and start drawing. Okay, I'm getting one breakout room set up, and please say something because I may be walking away to answer that phone call.
Professor, a quick question.
Yeah.
Will you be going over that white blood sheet or white blood cell sheet assignment? Or, I mean, it was pretty simple for the most part, but I was just curious if you would go over that.
How about we do that?
Because it's due today.
That reminds, it's due today?
I believe so.
We could do that too. I mean, it's pretty straightforward. I'm not going to go through the answers for each person's because I actually don't have your assignments right in front of me. But if you'd like, let me take a look at it to see what it looks like. I have to get out of here and go into, let me make that go away for a minute. Differential white blood cell exercise. I don't think there's that much you have to do on there, but I do have all the assignments.
I was just a little confused with like the first part where it was like state what condition you possibly have, I guess.
Yeah, so you're picking one from this row.
Right. From mine, I had eosinophils that were high, so I just put eosinophilia. Would that be like correct? Oh, okay. Okay. Oh, okay.
And then if it's abnormal, then you're going to look in here and give two conditions. So there was one group that someone was asking about this morning where you have more than one thing high. So then I would try and figure out what is common between those. And if things are low, remember you're not worrying about it because they're low only because it's a percentage. So when one thing goes up, the other has to go, some of the others have to go down. Is that clear what you're doing? Study this stuff too. Any other questions on that? Let me get chat going again. What the heck are you guys? Be close. Are you still there? Yeah, you are. You're just hidden behind another window. Okay, so let me see. Nothing else in chat. Is that good for going over that? I didn't catch what to label. I'm sorry. You mean what to label on the slides, Sarah?
Yes.
Yeah. So that study guide slide had everything that you were supposed to label. So if you go into the study guide, there was a wordy slide right before. Well, maybe I will go back in there again because I would like to show you something else from this. What you need to bring for next time or have for next time, for next Tuesday. So Sarah, this is the slide that lists everything you're labeling. The only thing I left off of this, it looks like, are germinal centers, which would be labelable on many slides, on the Peyer's patches, on the spleen, and on the lymph node. But otherwise, it's all listed here. So before I read it, did you get it?
You can't see it. No, I don't.
Oh, I'm sorry, I'm not sharing. Oh, God. There we go. I can see it perfectly well. Share the screen. This slide, draw all the lymphatic at 40, and then you're drawing 2 at 400. And what you're labeling is all on this, except for germinal centers. I would add germinal centers wherever you see them. It says locate germinal centers. It doesn't say label.
Oh, thank you. I was in the big study guide.
Okay. All right. Well, then when we come back on Tuesday, you're going to be doing the analysis for a lab that we would normally do in person called an immunodiffusion lab. Now, this is explained in the same lab exercise that we're in now. Whose number I can't read. Exercise 35. But what they do is something called an Ohlone test. It looks like Ouchterlony, but it's Ohlone test. And so there is a discussion about the advantages and disadvantages, or what the context that you'd use. The Ohlone immunodiffusion test versus the one that we are doing called a radial immunodiffusion test. And one of the things the radial allows you to do is quantify things. Ohlone identifies things, but radial immunodiffusion, you can quantify them. So you are going to be using data that's embedded in here from this radial immuno, that's what a RID test stands for. Here's some data that the students last semester gathered. And then you're going to eventually turn in a graph. Now, there's a lot of misunderstanding about this, how to go about doing this. So make sure you read the handout carefully. And then if there are questions about that, we can discuss them on Tuesday. So ideally read that handout before you get into lab, so you understand when I go through these slides. You have some understanding and also some questions if you don't understand how to do the analysis and to calculate the concentration of the antigens. Okay, let me see if there are any other chat questions. Maybe a new one, maybe it's just Sarah's. Okay. I missed something on the white blood cell we're designed items. I'm not sure what you mean by items, Dina.
Sorry, I'm confused, and I may have actually done it already. I don't know. I'm just
At the bottom, it asks you kind of two questions. It says, what condition do they have? So if they had high neutrophils, you would say they had neutrophilia. That's what you fill in. And then underneath, it says, what are a couple causes? Well, you look in that last column under neutrophilia, it gives you a couple causes of neutrophilia, and you put down 2 causes.
Okay, so I just have to find the actual
Handout?
yeah.
Okay. Well, I'm here, so you can always ask a question later.
Okay. Oh, that's what it is. It looks like we were given a group. And I
Oh,
think-
yeah, I only made a few variations. You don't have to work together in the group. I just
Oh, okay,
sorted you out so that, not everybody had exactly the same problem. And it also means that if you look at the other group's sheets, you can play with it on your own. You're only
Right.
turning in yours, but you could play with the other situation.
Okay, got it. And you did that. I remember you did that was the one you did alphabetically where
Right.
people with last name between L and, okay. All righty. I think I just forgot.
I know.
Sorry.
Dr. Kaufman? Ago and a week ago, really. It was a week ago. Yeah.
Yeah. Okay. Great.
All right. So by Tuesday, ideally read that red handout. And you don't have to read the study guide. We'll go through that, but read the red handout. Okay, guys, let me move you so I can close this window.
Dr. Kaufman,
here
are you
it is,
going to
the
create
red
an
handout.
assignment?
I'm sorry, what?
Create the assignment where we can submit the white blood cell differential.
Yes, submit it through the assignments. You don't see it on the list of assignments?
I think for me, I don't see either lab 9
Oh,
or
I may
10.
not have added it because I didn't write it. So I'll make sure that's there shortly. Okay? So you can go through assignments to submit it. Let me get out of here and go back to here. Stop share. And I will put it into assignments so you have a way.
Professor, it is over there. It says that with the white cell.
So why did you make
Oh,
it
yeah, it's in there. RID and immune exercise. Oh, no, lymphatic histology. Oh, white blood cell
Yeah.
histology.
The last line says.
Yeah, so the last line of that includes that assignment. So yes, go through white blood histology to submit your differential white blood cell count. Maybe I should rename that so then you know that it's in there. Can I rename? Doesn't let me rename it because I have to go into it and edit it to rename it.